Welcome back to the course. So we started our module 5, whose title was Immune Signal Transduction and Activation Thresholds. And in the last lecture we understood how this receptor signaling happens in B cell. And we decided that we'll discuss this B and T cell population differently. So in earlier discussion, what we have understood that there are immune cells, and these immune cells are recognizing the foreign molecules, and these foreign molecules is what is called antigens. And there are cells, particularly specialized cells such as B and T lymphocyte, which raised against those antigen in order to nullify the effects. And then further we started looking into how this signaling or how this activation do the phenomena in case of these B and T cells. So we discussed about B cell in the last class, and what was emphasized in the B cell activation is mostly the phenomena which decide the signaling is phosphorylation. So the phosphorylation of the receptor molecules or any of the substances which are present on the cell membrane after binding to the antigens can activate the signaling cascade. So we have looked into multiple different kind of kinases which are present in the B cell, and now we'll try to look into some of the things which are important for T cell. So as we know that these T cells are also very important because these are ultimately responsible for generating an antibody and also the cell-mediated immune response, because T helper cell or cytotoxic T cells are actually the cells which are involved in immune regulation for multiple different kinds of antigen. So let's move on and see that what are the things which are important for the T cell activation and signaling cascade. So when it comes to T cell signaling, these are important in terms of three different aspects. So the first thing is when the peptide which are present on the MHC molecules, which are again in turn on antigen presenting cell, encounters with a T cell receptor. So the first step is how these MHC molecules are binding to the T cell receptor. The second is the co-stimulation. What does it mean is when a cell is binding to the other cell or communicating with the other cell. For example, suppose this is one cell type and this is the other cell type. So this is cell number 1, this is cell number 2. Now they communicate with each other with the help of this, suppose the protein, which are called receptors. So the one cell can modulate the effectivity of other, or the one can also modulate the effect of number 2. But say this binding is not sufficient sometime, and therefore we need some more additional molecules from each side of the cell in order to start the process. So for example, these are another molecules which are presented from cell number 1, and there are other molecules which are presented from cell number 2. And now what they do is they strengthen this particular binding. So this binding was the initial binding, and the others are actually required to strengthen the binding, initial binding, and therefore these are called co-stimulation. So once they the co-stimulatory molecules, the other proteins are involved in the process, then we see that actually 1 and 2 are now involved in the basic biology. So this is very common in case of immunology, that there are proteins which are called co-stimulatory molecules, which we'll see in the next class. That what do you mean by co-stimulation? So one of the molecule which does take place in this process is called CD28. So that we'll try to cover in our next discussion. And the third one is once there is a binding of MHC loaded peptide with the TCR, which is T cell receptor, and co-stimulation, then the cells become active. And once the cells become active, they start secreting a lot of proteins. And these proteins are what we call cytokines. So this cytokine is actually supporting the proliferation of cell types so that this atmosphere become more homogeneous in terms of supporting the immune regulation. So therefore, this T cell signaling is controlled by three distinct processes. One is signaling towards the MHC binding. second is towards the co-stimulation, and third towards the further secretion of cytokines. So these are the three processes. Let's try to understand this again in a same way which I have discussed. So the signal 1 can be called as antigen recognition, and this recognition is again by binding of a specific peptide. This could be MHC1, this could be MHC2, because we are talking generalized about the T cell population, and we know that there are two distinct kinds of T cell, T helper cell and cytotoxic T cell. So these two can activate differently. This could be activating major histocompatibility complex 2, and the cytotoxic T cell can activate the MHC1 specific loaded peptide. So again, there could be a cross representation which we have discussed earlier, but in general these are the two cell population which behave differently upon binding with the different MHC molecules. Second is co-stimulation, which we discussed, and again, as I told that we will discuss it later, but since this we are talking about in context of T cell activation, we should understand there are molecules, some of the proteins called CD28, which are present on the cell surface, which can help to bind with other molecules such as CD80 and 86, which are present on the antigen presenting cell. So basically CD28 is the protein present in the T cell, and then there are other proteins present on the antigen presenting cell which are giving the loaded peptide and kind of sensitizing the T cell. So there are proteins such as CD80 and 86, and this binding can avoid the energy which we have discussed, right? So which is what we call functional inactivation. So only because of the co-stimulation only the cell can proliferate, so it can do the activity. Otherwise the cell will undergo energy, which is actually functional inactivation. So this is very important because we want to have a functional T cell or B cell population in the area where there is a requirement. And finally, the cytokines. We'll try to understand it in a pictorial representation as well. But cytokines such as IL-2 is what we have discussed multiple times, that it is a mitogen, a biological mitogen. That means it can help to proliferate the similar kind of clonal cell population in that area, and therefore we call same kind of clonal expansion. So this cytokine does play a very important role in order to proliferate the cell type and also to do differentiation. So once the cells are differentiated, then they know what to do. The particular cell, once they are differentiated, then their functionality become defined. Otherwise, these cells are considered as a stem cell. They don't have any distinct or specific function. Once they are differentiated, then they can have a distinct function. For example, if we are talking about any cells which are leading to forming pancreas in future, the cell is actually a stem cell. Now the cell particularly don't know what to produce, what not to produce, but when the cells are differentiated, then the cell become alpha cell, beta cell, or cells which can secrete different kind of hormones in pancreas. Particularly when we take an example of beta cell of pancreas, it is known to produce insulin, right? So once it is differentiated, then only we can say that the function of a beta cell of pancreas is to secrete insulin. Otherwise, the progenitor cell, the one which is mother cell, is not having any distinct role, and therefore differentiation is important. Therefore, the activation of T cell done in a series of processes. Once there is a binding, co-stimulation, and then the stimulation towards the cytokine can do a lot of other function. So this is what we have discussed overall. So this is a model of T cell activation which we have discussed. Now we can understand that this is an antigen presenting cell which is carrying the MHC2 loaded peptide. So once we are calling it MHC2, definitely this is a T helper cell population. So this is a T cell receptor, and T cell receptor is expressing a CD4. So just keep in mind that once it is CD4, this is automatically a T helper cell. When this is CD8, then we can understand that this is a cytotoxic T cell. So these are markers actually, right? So one marker can define the cell type. apart from that, we have a normal or the global marker for the T cell, and which is CD3. We will see it little later that CD3 is a very common marker for the T and B lymphocyte. However, these are expressed in other cell types sometime, but generally it's a global marker for T lymphocyte and B lymphocyte, CD3. But when it comes to T lymphocyte, the T lymphocyte can be very specifically called as T helper cell when they are expressing the CD4, and TC or cytotoxic T cell when they are expressing CD8. So this is what is lymphocytic marker. And just to correct a little bit the things which we have discussed, the B cells are having very specifically CD19 as a marker. CD19 is a marker for B cell, which we have discussed earlier, and this is regarding T cell marker. And second is co-stimulation. Again, this is CD80 or 86, which is coming from antigen presenting cell, while CD28 is coming from a T cell population. And therefore, once this is binding, there is a binding of this CD80 and 28, then this become more further enhanced to avoid the energy, the function inactivation. And once this is done, then we can see that the cells become stimulated, the cell become differentiated, and once these are differentiated, then we can see that a lot of cytokines are produced, and these cytokines in turn will bind to the receptor present on the cell type to activate the other processes. That means this could be an autocrine function. So the IL-2 is secreted from activated lymphocyte, and this can bind to its own receptor to do a same functionality in its own cell population, and therefore we can call it as an autocrine function, a self-activating function. So this is about the model of the T cell activation, 3 steps, and what are the receptors which are present in the cell in order to activate this. So let's move on and try to see that what are the receptors present on the T cell. So this is what is the overall diagram of a T cell receptor. So we are talking about a T cell. This is an extracellular space, this is a cytoplasm, and exactly like BCR, which is a B cell receptor, where there is an immunoglobulin chain, and then there was an Ig alpha and Ig beta. Here also we have TCR, which is called T cell receptor. There are two chains here, alpha and beta. Apart from that, as I told earlier, that CD3 is a common marker for T lymphocyte. So we have CD3 as a common marker, and this CD3 is available in different chain form. So there are epsilon gamma chain or epsilon delta chain. That means either we have a gamma or delta variant. What is important here is this TCR is fixed, so we have 2 chain alpha and beta. Apart from that, there is a tau chain as well. So alpha and beta is fixed in a TCR. Apart from that, we have a CD3 receptor, and this CD3 receptor has a common epsilon chain, while there is a variation in the other chain type, which is called gamma and delta. That means CD3 come in a 2 different flavor. Either they have epsilon gamma or epsilon delta. So we could see that these are the different forms of the T cell population, and there are variation in the CD3 receptor as well because of their chains. And these are the cytoplasmic domain of that protein, and each domain is containing this receptor called ITAM, which we have discussed in the B cell as well. So these are called immunoreceptor tyrosine based activation motifs, and these are actually getting phosphorylated because some of the kinase that will come into the picture little later will do a phosphorylation of the tyrosine residue present in this protein motif, and because of this phosphorylation we can see that these gets activated, eventually leading to further downstream signaling cascade altogether. So this is roughly about the T cell receptor and some of the proteins which are present on a cell surface of the T cell. And again, these are all transmembrane domain which are there spanning high in hydrophobic amino acids. Alright, so we'll move on and we'll try to see the 2 classes of the T cell receptor which I have discussed. So as I said that there could be a receptor where alpha and beta will be there, which can help in the binding of an antigen. So this is actually alpha beta T cell. or otherwise there could be a gamma delta T cell. That means they can bind to antigen very specifically in a different style. So we have based on the TCR protein, we can differentiate these two T cells into alpha beta or gamma delta T cell. Now, just to give emphasis towards the gamma delta T cell, these are the T cell which are involved in regulation. So these are T regulatory cell population, eventually leading to a lot of immune regulation in the mucosal epithelium. So these we will discuss separately when we will discuss about tolerance and that other stuff. So these are again the same protein like immunoglobulin. So we have a variable region on the top which can bind to the antigen, and therefore there is a variation in the actually antigen binding site. Then again we have a constant region which varies very less as compared to the variable region, which is really a hot spot, lot of amino acid variation, and of course the transmembrane region, and finally the cytoplasmic domain, which can do a cascading. So we in signaling we are mostly concentrating our discussion towards the cytoplasmic tail, because here this is the site where the proteins can dock and do a phosphorylation and other signaling phenomena in order to activate the T cell population. So these are the two type of T cell receptors. One is alpha beta and the other is gamma delta. So before going into detailed discussion about the proteins and what are the things, there are a whole bunch of different kind of kinases and also some of the molecules which are getting upregulated, downregulated, because in recent years the biology of immune cells or immunobiology in context of signaling cascade has been improved a lot. So our understanding towards the immune cascade, particularly the signaling pathway, has been increased to an extent that there are a lot of new molecules which are identified. There are a lot of different kind of pathways which are newly explored, some we are still studying, and there are hot areas of research where people are trying to look into some of the diseases which can lead to immune dysregulation or immunodeficiency. People are trying to look into the pathways. And by doing all this, people have identified multiple proteins which are involved in various different pathways. So I have just listed here for your reference. There is no need to remember all those things, but we should understand some of the functions of these molecules. For example, there is AKT, and of course this is called protein kinase B earlier. Now this protein kinase B, as itself indicate that it involves in phosphorylation. The variation could be many, so some of the cells can express AKT in a large quantity, some other cell type can express in the lower quantity. Phosphatase angiotensin homologue, which is called PTEN. This is again a very important phosphatase which can remove the phosphorus group and again activate multiple processes. Then we have this Janus kinase, JAK-STAT pathway, which we'll study when we will try to understand the interferon pathway. So Janus kinase is again when there is a kinase, that means it's a phosphorylating enzyme which can do a phosphorus addition to a protein, and STAT is the signal transducer and activator of transcription. So these are the transcription factors which are actually sometimes getting a homodimer, sometimes getting heterodimerization, and once they are getting dimer, then they are migrating themselves to the nucleus. Otherwise they stay back into the cytoplasm, and once they are going to the nucleus, they can do a transcription of the gene which are important for the activation of a cell type. Then we do have ADAP, which is called adhesion and degranulation promoting adapted protein, and BCL10, which is a B cell lymphoma, CARMA1, which is a card containing MAGUK protein 1, which is also called as card 11. So I'm just trying to give a brief discussion about all this, because these will be coming one way or other way in this course frequently. So I just wanted to highlight few of them. There is one which is called CRAC, which is a calcium releasing activated calcium channel. So calcium release activating calcium channels is actually a channel which can allow the calcium entry. So there are multiple processes which will happen in the cell which can allow the disturbances in the calcium homeostasis. In general, the storehouse of the calcium is endoplasmic reticulum in a cell. Once there is a problem in any sort of deviation in the cellular homeostasis, the calcium oozes out from the endoplasmic reticulum. And once it is coming out from the endoplasmic reticulum, it can activate a multiple pathway. Particularly in T cell activation, since we are talking about a T cell activation, there is a protein called NFAT, which we have discussed earlier also. This is called nuclear factor of activated T cell, and that's also regulated by the calcium concentration in the cytoplasm. Calcium is very sensitive in general because the concentration of calcium is always constant in the cytoplasm, and if there is any deviation, the cell does differently in order to maintain the calcium, say, concentration inside cytoplasm. And by doing that, it can activate multiple other kinase pathways which can lead to the homeostasis of the cell. Diacylglycerol we have discussed in B cell activation as well. The GADS, which is GRB related adapted protein 2. GRB2 is again a growth factor receptor bound protein 2. So again, these are all the protein which are present in the item region just to make more adaptation and strong signaling to the downstream activation pathway to the T cell. So I'll not go in detail, we can just go through each and everything because next figure can have all these protein one way or other way, so one has to understand that this does play a very important role. While there is a MAP-ERK kinase which is actually protein which can do a phosphorylation of the other kinetic protein. That means we discussed about the phosphorylation in the earlier classes as well, that once the protein is phosphorylated, it can do phosphorylation to other protein as well. So these are some of the kinases and some of the proteins which are important for this different kind of signaling pathways. This is not limited, there are many more, but these are very important for T cell activation pathway. Well, we will move on and we'll try to concentrate the rough idea about how this antigen presenting cell and the T cell activation takes place upon the binding of the antigen. So once there is MHC loaded antigen or the peptide comes in contact with the TCR, this TCR is getting activated by LCK, which is again a kinase. So we can see that these are present here, and these are again a molecule which can dock along with the TCR. So this can be CD4 or CD8 based on what kind of T cell we are talking, and this basically binding of the antigen can do LCK activation to do phosphorylation. So we can see in the second panel here that LCK phosphorylate the tyrosine residue which is there in the ITAM domain. So ITAM tyrosine get phosphorylated here by LCK, and because of this phosphorylation, the very unique protein JAP70, which is actually a zeta chain associated protein kinase 70, JAP70 here is coming and docking into the site. So if we can recall back in the last B cell discussion, we talked about the SYK, which is a spleen tyrosine kinase. So SYK kinase can be docked into the site post phosphorylation. Here instead of SYK, we are talking about JAP70. So JAP70 is a very specific zeta chain associated protein kinase. About 70 kilodalton size is coming into this site which is getting phosphorylated. And once this phosphorylation is done, it can cause further phosphorylation of linker for activation of T cell, which is again a molecule which are docked into the site of cell membrane. So linker for activation of T cell gets phosphorylated, and this phosphorylated form of LAT is actually recruiting lot of other kinases and signaling molecules exactly like B cell. So one of them is protein PLC gamma, phospholipase kinase, and then we have GRB2, which is again growth factor receptor bound protein 2, and SOS, son of 7 layers, which we have discussed in the B cell as well. So once we have phosphorylated LAT coming and recruiting this, we can see that this can do lot of different function, which we'll see in the next slide in details, although I'll not go in each and every pathway because that's not needed. But what is required here to understand that once there is a phosphorylation, this phosphorylation can lead to next level of phosphorylation, and by that all these events step by step can do some downstream molecular activation which can ultimately recruit the transcription factor to the nucleus. And once the transcription factor is moving to nucleus, they can do a gene transcription. And once gene transcription happens, then the T cell become activated to secrete cytokine, which is what the third signaling stage of T cell activation. So this in general, that once we have different kind of protein activated, then we can start getting the signaling of the T cell. So this is actually a T cell signaling cascade. So this is more complex and more cluttered slide. So I'll just try to focus on few things, and which is very important. So the first one we already discussed, that MHC2 molecules bound to the T cell receptor, and the T cell receptor getting phosphorylated by LCK. So we can see LCK is getting phosphorylated. After phosphorylation, the JAP70 protein is coming. JAP70 is again activating and phosphorylating LAT, and LAT is again recruiting a lot of other things. So and then ultimately what we are getting is this transcription factors such as JUN, FOS, AP1, NF-kappa B, REL, and NF-AT, which is a nuclear factor of activated T cell, is going to the nucleus. So here we have nucleus, and once they are coming into the nucleus, they can bind to their cognate areas in the gene which are supposed to do a transcription, and then we can see the production of messenger RNA to a large quantity, and this messenger RNA can go back into the cytoplasm, bind to the ribosome, and then synthesize a typical protein which will secrete out. So this is a complete, say, phenomena which is actually one has to understand that once there is a transcription, then what ultimately protein coming out into the system is actually governing the whole activation, clonal expansion, and other things. So two important aspects which we will discuss here. The first thing is calcium influx here. So we can see that this channel which we discussed, the calcium regulatory channels, these can influx the calcium inside, and the calcium can also come out from endoplasmic reticulum, the stored calcium, because we have activated certain pathways, and because of this there are proteins which are there in the cytoplasm called calcineurin, and this calcineurin is actually dephosphorylate. So they can do dephosphorylation. So we can see that the NF-AT, which is a phosphorylated form of the protein, is getting dephosphorylated. So the phosphate is going out and the NF-AT is coming inside. So there are proteins which are getting phosphorylated to go inside the nucleus, and there are proteins which are getting dephosphorylated. And NF-AT is one such beautiful example where the protein is getting dephosphorylated. So the phosphate is coming out, and then NF-AT is going inside the nucleus in order to do the transcription. So this is one. So just to remember that calcineurin is a calcium sensing protein in the cytoplasm. It can activate the NF-AT so that the NF-AT after dephosphorylation can go to the nucleus to do transcription. The second very important one is here. So we have certain kind of protein which cause actin polymerization. So actin polymerization is again a very fundamental process that happens in the cell, and particularly this is important when the cells needs expansion. They wanted to engulf phagocytosis, cell growth, extension, perturbation, anything which is related to the cytoskeleton, related to the cell proliferation is what is required, actin polymerization. Now actin is actually a cytoskeleton protein. So let's try to understand few basic fundamentals about actin protein and how it is getting polymerized. So actin is actually the mixture of G and F. G is called globular protein, globular actin. It's actually a monomer. So we have a single monomer. This is called G actin. And once these can bind to an ATP, it start getting an aggregation. And this aggregation can lead to the filamentous form, the bigger form of the protein, what we call the filamentous actin. So that means the monomeric form of the actin can form a filamentous form by the series of processes where ATP is required. So it's actually an active process where ATP binding is involved. There are three distinct steps here involved in the actin polymerization. And since we are talking about the dendritic cell, antigen presenting cells, we always know that these cells are having a lot of elongated structure in order to engulf the antigen. So we always see that the role of these is very critical, because they can form the lamellopodia or the philopodia in order to engulf the antigen. Apart from that, they can also form the structure of a particular cell type, and of course they can help to trap and engulf the pathogen or the entering antigen from outside. So the phagocytosis process requires larger surface area. For that we need actin polymerization. So this whole process of actin polymerization is divided into 3 stages. One is called nucleation, second is called elongation, and the third is called steady state. That means the combination of these 3 processes can lead to the production of filamentous form of the actin. So once this G monomeric form of the actin forms a nucleation, sometimes it is in the form of 3 or 4, so this can form the center, and more globular actin can come and form a small thread like a structure. So this is actually a nucleation state, where a nucleus, a small kind of a base, is formed, and based on that base each individual molecules are getting attached to it with the expenditure of ATP, and then slowly we see their extension, which we call elongation. And once the elongation comes to a point where the output and input is same, then it's called a steady state. That means the budding out of the globular actin and the addition of the globular actin is constant. That means the rate of formation and rate of dissociation is the same. Then we called simply the steady state of this process. So this is again done by multiple proteins. I have just listed 3 of them, and again we are not going in details. We just discussed this because this phenomena is important. So one is propylene, which again a very important protein responsible for ATP binding and stuff like that. Then we have ARP2/3 complex, and then formins. So these are the 3 proteins which are regulating this process, but ultimately the polymerization can do a lot of function in the cell which are listed here. They can involve in cell motility, either structure or endocytosis, which I told you with giving an example of antigen presenting cell. This is in a nutshell about the actin polymerization and why it is important in case of T cell activation. Well, we will move on and we will try to see another very important thing, which is called tuning. Now the tuning is important because tuning stands for once there is an activation or once there is an unwanted activation, we need to have some control mechanism. So some tuning of the T cell receptor biology is required. So body, in recent year it has been identified that there are many proteins which are getting upregulated or coming into picture when there is a requirement of tuning. So once there is a recognition of the self antigen, so suppose these are self antigen loaded peptides, then this TCR activation may happen, and this whole process can do a T cell activation. Now we don't want any huge amount of immune regulation against the self peptide. So there could be a lot of inhibitory pathway which we'll discuss. There are 2 checkpoint molecules which we'll discuss a little later. One is called CTLA4, which is actually cytotoxic T lymphocyte protein 4, and programmed cell death protein 1, PD1. These are the checkpoint proteins. So apart from that, the cell also expresses some protein such as CD5. Now this CD5 is actually a protein in recent year has been studied in great detail to do a tuning process in the T cell. So the T cell extra exaggerated response upon the binding of the self antigen or the self peptide loaded MHC is controlled by this CD5 molecule. So one can see that this CD5 can do inhibition. This is the sign of inhibition. So once there is a CD5 coming into picture, it can simply inhibit this process, and therefore there is a tuning of the signaling cascade. So 2 important points which are making CD5 different than these checkpoint molecules is they are constitutively expressed on naive T cell population, What do you mean by constitutively expressed? That means they are expressed at that time of requirement in the T cell, in the naive T cell. They are constitutively expressed, and their expression is quantitatively regulated by the signaling intensity. That means when there is more intensity into the cell or more intense P cell MHC molecules signaling, then we can see that there is more and more expression of the CD5. That means, so the tuning molecules are differentiated from the checkpoint molecules into two distinct aspects. One is their expression is constitutive, so they can express only on the naive T cell population when there is a requirement, and then their expression is quantitatively regulated by the TCR signaling. And what do we mean by the TCR signaling is, once there is more and more or intense TCR signaling towards the self-antigen, then we can see that the CD5 expression goes exponentially. Otherwise, they will have a controlled expression of CD5. That means the expression of CD5 depends upon how strong the signal is coming from opposite side, which is an antigen presenting side. More stronger the self-loaded peptide, more stronger the CD5 expression. This is what is the meaning of that. And these are present in T cell population, particularly in the naive T cell, which are not expressed or which are not exposed to the antigen, what we call naive T cell, which are not seen any foreign antigen. And these are the two molecules which we will discuss in our future classes. One is CTLF4 and the other is PD1. So just to summarize the whole study which we have discussed till now is what we understood, that there are two distinct kind of cells which are there in lymphocyte, T and B cell, and these cells are getting activated and upon the binding of an antigen, and once there is a binding, it can do a signaling cascade, and this signaling cascade can further lead to the expression of a protein. So let's summarize it into a very layman language. So suppose this is a cell, and this cell is, so this is a nucleus where you have a specific DNA, and once there is a, so this is a receptor of a cell, and once there is a binding of an antigen or any foreign protein, it can activate multiple protein partners in the cytoplasm. One such partner could be ITAM, and once there is a phosphorylation, then it can recruit many other proteins into this site, and once there is more and more protein docked into this site, it can do a signaling cascade which can activate many other proteins, and this protein is further going into the nucleus. And finally, since these are a transcription factor, they can bind to the specific DNA sequences, do transcription, and can form messenger RNA. And once this messenger RNA is formed, these are going into the cytoplasm to bind to the ribosome, and once these are bind to the ribosome, then we can see there is a production of a protein, and this protein is again coming out into the surrounding and do the effect. So basically, once there is an antigen entry, there is an effect coming here by this signaling cascade, and we talked about the T cell, we talked about the B cell as well. Now this whole effect, the activation of a protein, can actually nullify this antigen. This is what we are going to study in whole of this course. But this was particular, this activation of T and B cell is mostly towards how this signaling is transmitting the impulse from cytoplasm to the nucleus, and that is what is the T and B cell activation cascade. So in the next class, we'll try to look into how this co-stimulation working, and how these are regulating, how there is a nullifying response towards the self-antigen, and how this co-stimulation plays a variety of different roles in maintaining the T and B cell biology in a human being. Thank you.